Thank you. Good morning. Hi, I'm Charles Matthews, a principal at Clearview Healthcare Partners. We're a life science strategy consultancy that works for an array of biopharma, medical device, and diagnostic innovators. And I was asked just to give some prefatory remarks, hopefully to tee up what I think will be a really interesting discussion. Kate's going to give us some insight as to what's going on in terms of precision medicine, the FDA. And we've assembled a very prestigious panel in terms of folks that are able to weigh in on some of the key questions affecting our industry, such as laboratory developed tests. So I'll first, to start out, we'll just give you a little bit of context for where my perspectives are coming from. As I mentioned, we work for a range of innovators, and we focus on commercialization and market access challenges, and we base that off of a strong foundational understanding of science. We have a number of PhDs on our team. And within the diagnostics field, I work with laboratories that are developing innovative, often value-based tests, very often venture-backed. I work for IVD companies that are developing test kits and platforms. And I work for pharma that are developing targeted therapeutics that are trying to use these tools to identify patients for a response. And so if you think about that ecosystem, kind of all these worlds kind of crashing together with very different business models and unique challenges. So some of that relates to commercialization of products, thinking about what is the market opportunity for it, how do we successfully launch this. Some of it about how do we capture the value for it, how do we get paid, what code do we use, how do we define and articulate that value. And then a lot of that comes through the nexus of what I would call kind of hybrid projects of kind of looking at this from almost an investor's lens of if I'm about to acquire an asset, what is the value of that asset and how does that change over time given the evolving ecosystem? So it's an exciting place for me to be because I can see the way different companies operate, the way they prioritize different things. And so what I wanted to do is just kind of tee off of this discussion just by giving you a sense of some of the things that I think are some of the key developments, both in terms of reimbursement and regulation and how they are influencing and shaping the diagnostic industry. And then just in the slide after this, my last slide, I just want to give you a sense as to some of the key challenges moving forward. So starting on the regulation side, and maybe just in a broader context, I think we're in a very exciting era and a very exciting time to be in this space. We're seeing not only substantial progress in terms of technological innovation, in terms of developing new tools and exciting science showing ways of better understanding, predicting, and assessing disease, but that's being met with some new tools from the commercialization side. So thinking about different regulatory pathways and different reimbursement modalities. So the FDA has been very proactive in terms of thinking about new data and evidence concepts. I think one of the challenges we faced is with something like NGS, it's hard to go through the usual pathway of I'm going to validate every single individual biomarker on the same level with the same type of information when that may not be possible with a 500 gene panel. So some of the recent approvals in terms of NGS are showing ways of kind of looking at everything in a more comprehensive way. And a great example of that is things like these PanCancer approvals, where we're looking at approvals based on mechanism of action or driver mutations as opposed to individual specific data within a particular indication. And that is particularly true as you now say we've actually made a lot of progress in terms of addressing cardiovascular and rheumatoid arthritis, kind of large category diseases, and now increasingly the pharma industry is focused on rare and orphan disease and subtyping patients and trying to identify approaches for that. That creates a novel and additional evidence burden challenge. And one of the tools that the FDA, and Kate may speak to this, has been thinking about and trying to process is real-world evidence development. So are there ways to bring products to market and then continue to grow and develop evidence over time as it's used in practice, as opposed to this kind of one-way pathway of like you get approval and then after that, who knows what happens. So that's exciting. And then also we have multiple methods and sample types being explored. So a lot of the talks at this conference are around multi-omics and different methods of not just going beyond sequencing, but thinking about proteomics, metabolomics. A lot of innovation in terms of sample types, thinking about not just tissue-based analyses, but liquid biopsies and different tools. And one of the interesting things to me is some of my diagnostic clients now are not really in vitro companies. They're maybe in vivo companies or they're strictly data players where they just have a bunch of data and they analyze it, they come up with a unique algorithm. And the FDA is thinking about that as well and saying, okay, there's a pathway for digital health technologies to achieve therapeutic approval. So that's an exciting way to think about the value of diagnostic tools changing over time. Maybe it's not just about what goes on in the test tube, but it's about the data and the information being provided. And there is a pathway for that being created. And the last piece on the regulation side I want to touch on, I know there's a focus of the panel, is this kind of role of LDT versus IVD. And I know that different individuals have different opinions of this. I do like the fact that we have both of these pathways. I think it provides some different options in terms of ways to commercialize. I think what we need to do maybe is just get better about thinking about when is the right pathway used in place for different specific use cases. And I'll talk about that more in a moment. On the reimbursement side, we had a seminal event, which in terms of next generation sequencing, where Medicare and FDA collaborated to create a parallel review process for next generation sequencing. But then quickly after that occurred, we ran into some implementation challenges. So there were other talks earlier in the conference about BRCA and potential need for reconsideration about maybe unintended consequences of some of the language of that. And then we also still deal with a challenge with an area that I focus a lot on, which is commercial payers and trying to get their minds around what's the value of next generation sequencing on an ongoing basis. And then we also have some exciting innovation in terms of PLAs. That is a kind of a new coding category that allows individuals to describe their assays as specific to their technologies. And that provides some unique flexibility in terms of if I have something I think is unique and proprietary, I can go get my own code for it. I can navigate different payment pathways. I can define and articulate a value proposition that's not wedded to the methodology of the assay. And the last piece, and this is an important piece, not at this year's, but last year's talk, Next Gen DX, there was an interesting talk by an investor in this space. And he said, one of the fundamental challenges for me investing in diagnostics is the ROI model. It's understanding, okay, how am I going to get a return on this investment? Some of these regulatory and reimbursement challenges are front and center in their minds. The other one is, what's the exit look like? And so here we've seen some really interesting things where I think it has the potential to maybe reshape some of the investor philosophies and thinking. So you think about a company like Exact Sciences goes through the trouble of securing parallel review and securing FDA approval. It leads to a large market cap, and they actually had so much market cap they were able to acquire Genomic Health, which I'm still puzzling about how exactly the combination of those two fit together, but $2.8 billion is a big number in the diagnostic space. And it's exciting to me to see that occurs, and hopefully two one-trick ponies can now come up be something more than a two-trick pony, but we'll see. And then the other activity I want to mention is it's related to the PanCancer piece. Is the pharma industry for years was dragging and kicking their feet saying, we not only like precision medicine, but they don't really like precision medicine. They would love to have all comers, and they would love just to say it works with 30-40% efficaciousness across the board. But now I'm starting to see, particularly some of my startups, they're looking at a story like Loxo Oncology and saying, boy, there's a unique confluence of events here where if I'm able to identify a targeted driver mutation, expedite regulatory approval with a very targeted study with not a huge number of patients, it can lead to a substantial valuation. So Lilly snaps up Loxo Oncology for $8 billion. So now I have a number of pharma players that are looking at that and saying, boy, how can we duplicate that story? So I think we're on the tip of, if these different factors in terms of reimbursement regulation combine, then it could potentially foster more examples like this. So the last thing I want to leave you with is, it's one of the things I love about this conference is there's so many different tracks. I'm interested in all of it. I work in infectious disease and oncology, et cetera. And the challenge is like, well, how do we kind of relate all this? Are there things that we can be learning from one side and applying to the other? So I think holistically, one of the big challenges that I see is the value of more information. That's what we're struggling with as a diagnostic industry. How do we talk about the value of what is the value of doing multiplex oncology testing or infectious disease? I'm going to tell you about 50 pathogens as opposed to 2. And what does that mean for care? And I think it's a classic conundrum because we haven't had the technologies to assess that previously. So now we have the technology. Now we have to take that next step of defining, articulating what that looks like. And that is not only in terms of at the initial point of diagnosis, but then what's the value of more information from that point moving forward? There have been a number of talks here at this conference about the potential applications in ongoing monitoring, be it minimal residual disease or tumor burden analyses, different ways of kind of looking at patients over a period of time and saying, is the treatment working, not working? any type of monitoring technology faces this challenge of how do we define and articulate the evidence that supports the use case for ongoing testing of patients. I could test them every week now if I wanted to. What would be the value of that? So I think that's a central question, and we're going to have to navigate that. The other one is, on the right side of this diagram, is thinking about the feasibility and practicality of all this. You can say, I can develop a perfect solution, and some of the solutions I have seen at this conference have been very exciting in terms of combination of an imaging approach with a sequencing, with a tumor environment, with combining 12 different modalities. If it's only available in an ivory tower that's an academic medical center that can be the only one and only site on earth that's able to do this, then it may not have its full benefits. So I think there's another vein of innovation we're continuing to explore that's around how do we potentially blend the evidence development around research and how we're tracking the value of the information we're providing is actually learning more about the disease, it's not ongoing care. And then there's also how do we turn that from a translational science perspective or tech transfer perspective into practical and applicable tools. Like what's the version of this that can be used every day out in the community? And that's a big challenge, and I think about some of the exciting things in the point of care track, talking about kind of moving testing closer to patients to avoid some of the practical barriers that have people limit their potential options. And the last thing I want to touch on, and this is a key focus of this panel moving forward, is around accuracy. And how do we define what the right testing approach is? And do we really know what accuracy is? And I will argue that the FDA has a viewpoint that's focused on accuracy as it relates to the test kit for things like GMP. But even if the test kit that leaves the factory is perfect, then it lands in the lab, and the lab has to do a good job with it. So are we sure that we're getting accurate testing from the lab perspective? And what are we doing to make sure the LDT versions, or versions that are fundamentally kit-based but still have lab-to-lab variability, are being done correctly? So that I think is going to be an underpinning to a lot of what we need to do moving forward. And hopefully the panel will provide some insight as to how to balance that moving forward. What I'd like to do now is introduce Kate Donigan, who's kindly joined us from the FDA. Kate is the Acting Deputy Director of Precision Medicine and Molecular Genetics in the FDA's Office of In Vitro Diagnostics. She's earned her PhD in genetics from Yale University in 2011, where she focused her studies on tumor-associated mutations in human DNA repair genes. She completed her postdoctoral work at the Laboratory of Genomic Integrity at the NIH Child Health and Human Development Department. And she also was in a fellowship program co-sponsored by the American Society of Human Genetics and the National Human Genome Research Institute. So she served as Congressional Fellow at the Office of Senator Elizabeth Warren, and she also joined the Office of IVD at the FDA in 2014 with a focus related to diagnostic regulation and personalized medicine. So we're honored to have her share some of the things that are going on in the FDA.
Good morning, everyone. Thank you. So as Charles mentioned, I'm Kate Donigan. I'm the Acting Deputy Director for Personalized Medicine and Molecular Genetics in the Office of Vitro Diagnostics and Radiological Health, or OHT7, as we are now known. And today I'm going to be giving an overview of where we are now and where we are going in the future. So I just wanted to provide a brief overview of our office activities to highlight that we really have always taken a total product lifecycle approach to everything that we do. And by that I mean we do the pre-market side, we issue relevant guidance documents, we also have the third-party review program that is run out of our office, and we also do the post-market side, so the surveillance and compliance activities, and a big part of what we do in my group is community outreach. So our TPLC approach was so successful that the entire center has adopted the TPLC approach for our new super office, which includes 6 other OHTs, which were derived from the ODE side of the house. So we are all now one big super office in CDRH. So as you can see in this very convoluted diagram, we have a lot of numerous and varied activities that we do in the office. And so, as I mentioned, we have the pre-market side, we have the post-market side with surveillance and compliance. We also do CLIA-related activities with CLIA waivers and CLIA categorizations. And we also have the rad health side of the house. And additionally, my group really focuses on the policy side. So we do a lot of research into policies that we could focus on for really novel and upcoming technologies. And so I will talk a lot about those activities specifically. And as I mentioned, we do a lot of outreach activities, public media requests, that sort of thing. And so it's a big office. It's the biggest office in CDRH at the moment. And the TPLC approach has really worked well for us. So I just wanted to go over some 2018 highlights to give you a very broad overview of the activities that we have done in the office in the last fiscal year. So we have, in fiscal year 2018, approved 106 novel devices, which is actually a 40-year record. We finalized 2 NGS guidance documents, which the personalized medicine group was really involved with, and that was the use of public human genetic variant databases to support the clinical validity of genetic and genomic-based IVDs, as well as the the final guidance, which I'm just going to refer to as the Analytical Standards Guidance because it has a very long title, and I will talk about both of those guidances later in this talk. We also approved the first fully implantable device to measure glucose in people with diabetes, as well as an insulin dosing system expansion to include patients as young as 7 years, and this was a really big breakthrough for us. We also published a plan around third party. So we know that the third-party program has some challenges, and so the guidance that we published here really is focused on eliminating the re-review of third-party reviews by FDA. We also granted the first DNA-based test for minimal residual disease for hematologic malignancies, and this was an NGS-based test. We granted the first brain injury marker test, and we also had multiple breakthrough device designations. So year to date, in fiscal year 2019, we've published several draft and final guidances. Just to highlight here, we have a dual 510K and CLIA waiver guidance that was published in draft. We have a final coordinated development of antimicrobial drugs and antimicrobial susceptibility test devices guidance. A draft developing and labeling IVD for a specific group or class of oncology therapeutic products. That's known generally as the class labeling guidance. And that was in draft, and we are working to finalize that. We also published in draft a self-monitoring glucose test system for over-the-counter use, as well as blood glucose monitoring test systems for point of care. So a lot of work in the diabetes space in this year and the previous year. Additionally, we finalized the Class 2 Special Controls guidance for bacillus detection. We published a final rule to exempt flow cytometer instruments, and we also, I'm sorry, that should be on a separate line. We also recognized the first genetic database, which I will talk about later. Additionally, we granted a few really key de novos this year. The first interoperable insulin pump, the first mycoplasma genitalium assay, which we know there was a really big need for in the public health space. The first FISH assays for chromosomal abnormalities for patients with hematologic malignancies, as well as the first human milk analyzer. So a wide variety of things that we do in the office. Now I'm going to talk a little bit about the recent increase in breakthrough designations that we've been seeing. So just to refresh everyone's memory, which I'm sure everyone in this room knows, the breakthrough designation criteria must provide for more effective treatment or diagnosis of life-threatening or irreversibly debilitating human diseases or conditions. So you essentially have to meet at least one of the following criteria. You have to represent a breakthrough technology. There has to be no cleared or approved alternative. You have to offer a significant advantage over existing approved or cleared alternatives. So if there are existing cleared or approved alternatives, and you can demonstrate that you are better than those alternatives, you might be able to get breakthrough designation. And the last criterion is the availability of which is in the best interest of patients, which is really the focus of this entire program. So we've been seeing, as I mentioned, a really large increase in breakthrough devices coming to market through FDA. We have over 156 designated devices, and 50 of those are diagnostics. So, around a third of all the breakthrough designations that come to CDRH come to OIR. We've authorized 11 devices, which includes 6 PMAs, 3 510Ks, and 2 de novos. And just some early successes in this program to highlight. We have the Foundation 1 companion diagnostic, which was the first breakthrough to market in the diagnostic space. It was also the first PanCancer companion diagnostic oncopanel. As I mentioned earlier, we authorized the first breakthrough de novo, and that was the Banyan Brain Trauma Indicator. It's the first blood test for traumatic brain injury. And there are many more to come in the pipeline. Next, I'm going to talk a little bit about the 510K third-party review program. And we really believe that this program serves to improve the public health by allowing us to leverage the expertise of third parties and efficiently review 510Ks in a way that provides a vision for focusing our resources. So we are able to focus our resources on the higher risk and more complex devices that are not in the 510K path. We are able to maintain confidence in our third-party reviewers. So our third-party reviewers review these lower risk and less complex devices, and we essentially just review their memo that they send us. And we really think that this process enhances the speed to market because it's a voluntary alternative to FDA's 510K review. So we look for this process to decrease FDA time, to increase the review quality with our third-party partners, and reduce the total time to decision. So I just want to point out that this is our list of accredited third parties, and we have a relatively new addition to this list, the New York State Department of Health. So New York State reviews lots of tests for patients that are tested who reside in New York State. And when we authorize the Memorial Sloan Kettering MSK Impact test, we leveraged their New York State package, and I will talk about that a little bit. But at the same time, we also accredited New York State to be a third-party reviewer for not only tumor profiling tests, but lots of other tests in different areas as well. So if you go to this website, you can actually pull up the entire list of tests that New York State is authorized to review for us. It's quite lengthy. We do acknowledge that there are some challenges with our third-party review program. So we have seen in the past that there are poor quality submissions that often require our re-review, which really decreases the value of the program in our mind. We also know that there is limited public information that makes the review difficult. So 510K third parties require the use of public information instead of clinical data, and that's definitely been a challenge in the IVD space. So we really thought, we need to strengthen this program and make sure that we are using it to its fullest extent. So we issued a draft guidance on the recognition of third parties and outlining a strategy so that we don't have to routinely re-review these things. So that was released on September 13th, 2018. And we also have a commitment to increase the program staff from 1 to 5 FTEs. So this should really help. This is the draft third-party review guidance I just mentioned. So the key for IVDs in particular is that it really adjusts the restriction on clinical data review in the context of third party. So we have the statement here in the guidance that says if it contains simple clinical data, such as simple clinical images or tests using banked specimens, then it may be eligible for third party review. And that would mean that most IVDs are actually eligible for third party. Next, I'm just going to touch briefly on this concept of collaborative communities. So we recently launched a website for this concept of a collaborative community, which outlined here is our vision for collaborative communities are forums where public and private sector partners, including FDA but not run by FDA, can work together on medical device challenges to achieve common objectives and outcomes. And these collaborative communities, in our vision, would be convened by interested stakeholders, not by FDA. And these communities could exist indefinitely and produce deliverables for regulatory input as needed and tackle broad challenges. So one of our strategic plan objectives is to, by the end of 2020, have formed 10 of these collaborative communities. And if you go on this website here, you can actually download, there's a brochure that talks about how you might form a collaborative community. So there's actually a lot of really helpful information that we've just posted on our website. Finally, getting into more of the area that my group focuses on. So I just want to highlight that FDA has a very long history of enabling precision medicine. We have approved 37 companion diagnostics, cleared or approved. We have also approved over 100 nucleic acid-based tests, cleared or approved. And we've also issued more than 24 guidances on these topics since 2005, and more to come. We also have many successful companion diagnostic examples in the area specifically of oncology, where most companion diagnostics are currently found today. So we have 79 approved IVD companion diagnostic therapeutic product pairs. We have 36 approved IVD companion diagnostics in the field of oncology again. And we also have 30 approved cancer therapeutic products. And if you look at the way these numbers match up, you can tell that we have approved several IVDs that actually are indicated for multiple drugs. So this is very exciting because you could essentially order a single test and get information about the use of multiple drugs for a patient. I also want to touch a little bit on our work on oncopanels. So, this diagram here can be found at our fact sheet on the website I've indicated below. In this fact sheet, this was following the MSK impact authorization. We laid out what we believe is a clear, defined pathway for these types of tests to report cancer mutations for both tumor profiling indications and companion diagnostic indications. So the pathway that we've outlined in the fact sheet goes over the different levels of evidence that you would need to report out the different tiers. So the third tier here, the second tier, and then the top tier is companion diagnostic indications. So using this model generally, we have 3 key submissions that we authorized. So we have the Thermo Fisher Oncomine test, the MSK impact test that I've referenced today, and Foundation Medicine's Foundation 1 CDX. And that, as Charles mentioned, was part of the parallel review program with CMS. Now moving on to FDA's general vision for how we would regulate NGS-based IVDs. And one of the things Charles mentioned was that there's essentially no way to validate every single variant in a whole exome sequencing test, for example. So we've really focused our attention on the need for technical and analytical standards in the area specifically of germline diseases. That's where we started. That seemed to be the most straightforward area for us. So we described a potential expedited path to market for tests for inherited germline diseases that are suspected, and that the guidance that I mentioned, the analytical standards guidance, really outlined standards that we would like to be developed by the community, and that we could recognize the standard so that the community could update the standard over time, and that we could continually recognize the standard. So it is a lot easier to evolve a standard than it is to evolve special controls. We also had a second guidance about clinical databases. So we really hope that FDA recognized databases could be used to support clinical evidence as a source of valid scientific evidence for genomic-based tests. So our goal is that test developers could use these databases as sources of information to support the link between genetic variation and disease. And our hope would be that such databases could be used in lieu of traditional clinical studies, where appropriate. Additionally, as part of our efforts in the NGS space, we are looking to develop and characterize and make publicly available NGS reference sample sets that could be used to support the validation of these tests. So we've been doing a lot of work on somatic reference samples recently, and I will talk about that at the end. But essentially, we want to make available to the community fully characterized samples and DNA sequence and generate an evolving truth sequence. So as I mentioned, the genetic database guidance that we issued, the scope of that guidance is publicly accessible databases of genetic variants. If the database is private or proprietary, it is not in the scope of this guidance. The guidance includes recommendations for administrators of databases to demonstrate that the database could be considered a source of valid scientific evidence to support the clinical validity of NGS-based tests. This pathway, I want to emphasize, is voluntary. So we think of it as similar to a standards recognition pathway. We also think that evidence from these databases, as I mentioned, could support the clinical validity of future NGS-based tests. So the question that we get all the time is, how could IVD sponsors actually use these things? And so the goal of the program is such that assertions in these databases could include a variety of variant types and descriptive language, but we really review the SOPs that the database uses to generate that evidence. And we also need to point out that FDA recognition, as described in the guidance, does not extend to treatment recommendations. So the database recognition process, if you run a database and you are interested, first begins with a contact us, let us know that you are interested in recognition. You can contact us at the email address that I'm going to provide at the end of the slides. So we provide you advice before you actually submit your database for recognition. We help you kind of structure your submission and tell you what we would be looking for. Then the third step is to actually submit your formal recognition application, and that is done through an informational meeting Q-sub process. And then finally, we review the applications, and we can request information interactively through this informational Q-sub process and issue a decision on recognition. And step 5, hopefully, ideally, is possible FDA recognition. So last December, we actually recognized our very first database. This was very exciting. This was the ClinGen database run out of NIH. So what did we review when we recognized this database? As I mentioned, we reviewed their SOPs, including their processes and validation studies for variant evaluation, data integrity and security, and made sure that all the evidence underlying the assertions was fully transparent. We also reviewed the policies for how the consortia qualifies and approves people who do the curation, including their conflict of interests and disclosure policies. And then we actually did what we refer to as a spot check, where we took their SOPs and followed their SOPs through the process to make sure that we arrived at the same result that they did. So that is just our sort of validation process that we go through to make sure that the SOPs are actually being applied appropriately. So ClinGen has now been recognized as a source of valid scientific evidence that could be used by IVD developers to support clinical validity in pre-market submissions in certain contexts. So we think that developers of genetic tests will have the assurance of the reliability of the data in this database, and that they can use that to support their marketing authorization rather than continuing to generate the same data on their own over and over. And the scope of this recognition is really important to emphasize here. The scope of the ClinGen recognition is germline variants for hereditary disease where there is a high likelihood that disease or condition will materialize. So essentially high penetrant conditions. And the decision summary, I would encourage you, if you're interested, is listed here at this link, where it's extensive and goes into everything that we looked for when we did this recognition to give you a sense as to what we would look for in an application. As I mentioned, we also had the analytical NGS guidance, which is how I'll refer to it. It has a very long name. The scope of this guidance really applies only to targeted or whole exome sequencing NGS-based tests that are intended to aid in the diagnosis of suspected inherited germline diseases or disorders. So in the guidance, it's really an analytical standards-focused guidance where we make a series of technical recommendations for how NGS test developers can design and validate their tests. We also really tried to make it a broad guidance that accommodated a variety of test designs, components, and different indications within that broad indication. It also talks about validation, what the performance characteristics of your test should be, and what different evaluation studies you should be looking to perform. We also included labeling recommendations, and the entire purpose and goal of this guidance was really not to form the basis of special controls in a submission, but to really drive the development of the community around a standard such that we could recognize that standard and the standard could evolve over time. So hopefully Such a standard will be developed, and there are several in the works. But really, we hope it would form the basis of a future FDA-recognized standard, and in the absence of that standard, it could be the basis of special controls. And the guidance actually also discusses a potential expedited path to market for tests that meet these standards. So it goes into potential 510(k) exemption process. We also, as part of our personalized medicine efforts, still have Precision FDA, which is our collaborative omics platform. So it's essentially a sandbox environment where you can make an account for free, log in, use data sets and reference samples that are already loaded onto the platform. You can have your own private space. You can have a public space where you can share with others. And so we have a lot of activity on this platform where people are really interested and excited to join their colleagues and collaborate in this space. We also, as part of promotion of Precision FDA, have launched several challenges. So these challenges are really to engage the community and get people onto the platform to really understand and realize the utility of the platform. So we have a couple of different challenges highlighted here. We have Truth Challenge, Consistency Challenge. So these are essentially challenges where people put their data on the platform, run their bioinformatics pipelines, and submit their results to us, and we present folks with awards. So the whole purpose of Precision FDA generally is to really drive regulatory science in this space. It is currently not being used for any regulatory purpose. We are also involved in the generation of reference samples. So our goal in this space is to develop, characterize, and make publicly available NGS reference sample sets for a variety of uses. And this is really a community effort that has brought together lots of different stakeholders. And so the goal for using Precision FDA for this effort is to deposit these reference sequences on this easily accessible platform for anyone to use, and to integrate all the calls and develop a constantly evolving truth sequence. And we have characterized samples available, and we would like there to be sequence and metadata available as well. So again, another plug for Precision FDA. If anyone hasn't made an account, you can go on and request one and participate in the program. This just highlights the variety of reference sample projects that are going on that we are involved in. And there are lots of differences and similarities in the different programs. But as you can see, it's really a community-driven effort. And one of these efforts that I just want to highlight is an effort out of the National Institute of Standards and Technology to kind of make folks aware that CDRH has actually supported the NIST Genome in a Bottle reference material development since 2012 through various IAAs and other mechanisms. And so through this Genome in a Bottle effort, we have developed 5 reference materials at this point, everybody's favorite NA12878, and then followed up with the various trios, and there were microbial genomic DNA standards as well. Moving on to MDIC. So the Medical Device Innovation Consortium is a public-private partnership that was created to advance regulatory science for medical devices to benefit patients. So it brings together federal partners of FDA, CMS, and NIH, as well as industry and various nonprofits focused on patients, providers, and academics. The MDIC Clinical Diagnostics section fosters innovation by speeding patient access to new IVD tests, by developing new tools and methods to improve processes to assess the safety and effectiveness and the value proposition of these tests. So there are lots of different aspects involved in the MDIC Clinical Diagnostics section. Includes things like the fingerstick blueprint, the IVD real-world evidence framework, the clinical evidence framework, and the surrogate sample network. And the somatic reference sample effort is, I'm going to talk about that shortly. And we also have SHIELD, which is the systematic harmonization and interoperability enhancement for lab data. The cancer genomic reference samples, aka the somatic reference samples, or SRS, is an effort we are running through MDIC to improve the efficiency and cost effectiveness of accurate NGS-based test development and validation. And this is really a collaborative effort, and we've heard time and time again from specifically the oncology community that there is a strong need in this area to develop these reference samples. So the benefits of our work and the work of MDIC broadly in this area is that these reference samples would be available to aid in efficient NGS test development and validation in the somatic space. So hopefully the goal would be to streamline and obviate steps in the regulatory process for diagnostic test developers, provide transparency about test performance, and compress development timelines for targeted therapeutic development. And again, this effort for somatic reference samples involves several FDA collaborators, some of whom are here today, the MDIC collaborators, and really this is an effort to just make these samples publicly available and streamline the process. So there are two phases of this project. The first phase is to publish a detailed description of the prioritized set of samples and variants. So as you can imagine, there are lots of things to pick from, and you really have to prioritize where you're going to start. There is also a plan for the development and characterization in phase 2, and MDIC will determine to continue to phase 2 or transfer to a third party within a year. Phase 2 would involve contracting and overseeing the production and characterization of phase 1 identified high-priority oncology NGS reference samples and transfer the outputs to interested commercial entities. Again, this is a public-private partnership. And these project outputs would be made widely available to allow the scale-up production and broad public accessibility by interested commercial entities. And that concludes my talk for today. As I mentioned, if you have any interest in any of the things that I've discussed today, especially the database recognition program, please feel free to email me or to email the personalized medicine group at FDA.